Now it's 4 minutes past 5, so I think we are good to go. Um,
Yes, it
I'm gonna
starts.
I'm gonna hand this over to Gotam. Gotam has prepared a lovely introduction. So he will introduce the guest speaker. We are super excited to have Jordi here, and more details come now from Gotam. Thanks very much, everyone.
Yes, thanks everyone for joining us. Today we'll have the pleasure to listen to Jordi Garcia-Ojalvo, who actually hails from a background in statistical physics, in which he obtained his PhD at the University of Barcelona back in 1995. And during his first two postdocs in Atlanta in the US and in Berlin, he was working on laser dynamics and noise effects in excitable media and neuronal systems. And he switched to systems biology only around 2003 when he was a visiting professor at Cornell University. And he currently holds the following titles. He's the chair of the Department of Medicine and Life Sciences, and a full professor at the University of Pompeu Fabra. And he also leads the Dynamical Systems Biology Lab there, where they do a lot of cool research. I invite you to check out his page. He's also a visiting researcher in biology at Caltech since 2006. And further, he's also an executive board member of the Spanish Royal Physics and the Biophysics Societies. We are very excited to have him here. He will be talking about, or he will argue about how he thinks cells model their complex time-dependent environments through dynamical internal representations encoded in their molecular repertoire. So it sounds quite fascinating. The stage is yours, Jordi. Thank you.
Okay, wonderful. Thank you so much, Gotham, and also Leo for the invitation. It's really a pleasure and a privilege. I can recognize some of the names among the participants, not everybody, but anyway, hello to everyone. So this is really very exciting. Thank you again. Good. I also like very much the topic of this group, and I didn't know it until you contacted me, and I only recently, basically today, I asked to join the Google group. So I hope that I will join you in future talks because the program looks really great. Apart from my talk, of course, this is just a minor thing. Okay. Good. So what I wanted to talk is what the things that we have been thinking about in recent years, me and other colleagues, about the idea of how cells are seeing the world, if you will. How do they see the world and how they adapt? No? And I'm talking about cells, but I would like to, of course, to start, since this is it's very interesting that the name of the seminar seminar series includes the idea of looking at this kind of adaptation in neuronal systems. So it is a fact. Let me see if I can click on the slides. It is a fact, or I think we all agree that adaptation is basically one of the main characteristics of life. So we can say that adaptation covers all the species that are on Earth. They adapt to the environment, and this is what distinguishes from rocks, for instance. So, but on the other hand, there is this, if you will, dichotomy. If you look at the tree of life, which of course it's completely, it's a misleading picture to view it in this unidirectional way, but it doesn't matter. But if we look at it like this, there is this dichotomy between what happens at the top, quote unquote, of this tree and what happens at the bottom. No, at the top we believe, look, adaptation is driven by brains and neural systems, and these animals and higher organisms have these complicated ways of adapting to the environment and highly complex. Whereas at the bottom, maybe when we talk about bacteria or other unicellular eukaryotes or simpler organisms, if you will, well, maybe the adaptation mechanisms are simpler. There are maybe smaller circuits, and in some cases we know a little bit how they are made of and how they are built. But there is this dichotomy in terms of complexity, if you will, of the mechanisms and how we view the mechanisms. And what I would like to claim is that maybe there is not such a dichotomy in some sense. And this is something that has driven my lab for quite a few years. This analogy in terms of, at least from the functional level, the needs are similar, right? I mean, brains receive these multiple sources of information that are that are dynamic. Sorry, I'm trying to also, by the way, somebody has a question, either raise your hand, or I also have the chat here. So I'm trying to monitor this thing. So anyway, so on the one hand, we have on the top of this, on the quote unquote top of this tree, we have brains dealing with these complex environments in which you have multiple sensory signals which are temporal. But of course, functionally, the same thing is true for the quote-unquote bottom of the tree of life, or for instance, the level of single cells. These single cells have to cope with exactly the same challenges. Again, there might be a difference in the complexity, but still, you have multiple signals that these systems have to integrate. These signals depend on time, so therefore there is an environment which is spatio-temporally complex, if you will. So the question is, how do cells respond to this? And I always like to start with an example for why is this the case and why is this relevant and why the environment is really not so trivial, even at the level of single cells. And the example that I like to mention is, for instance, our cells. Any kind of multicellular animal such as us, in our case, we are made of 5 trillion cells, more or less, but we all begin from a single cell. And of course, the process of going from one cell to trillions of cells is very well established, and developmental biologists, some of you may be already in the audience doing this. I mean, it's a very interesting process from which we go from one single cell to trillions of cells. And of course, in that period of time, the cells have to decide to become different cell types at the right time and at the right position in space and react to this. So this is very well known. And right now, I would like to use this as an example, and I would like to focus on a stage around this point. I guess you can see my pointer here. This is basically the point at which form starts to appear. No, there's some kind of morphogenesis. Also at this point, if the number of cell types is also starting to explode. From just a few cell types, you have many more. And then I would like to focus on this. And I would like to show you a video by Kate McDowell that she published a few years ago from the early mouse embryo around this stage. And there you can see how the system evolves from a situation in which you have cells in a more or less in a clump to something which already has some form. Here in this video, you cannot see the cell types, but of course here you have multiple cell types. So what I would like to discuss here is, okay, if you look at that one cell, you can pick one cell at random, and you look at the local environment of this cell, both as time evolves, and also in comparison with another cell somewhere else, you can see that the environment is very different in two cases. The environment changes continuously in terms of what are the neighbors, what are the mechanical forces that this thing is feeling, what are the cell types and the different cellular chemical composition of the cells, or chemical decisions or chemical state of the cells around them. So this means that the environments are very different from different cells, and they are all changing time. Now, it is hard to believe, even though one possibility of how this happens, and I'm going to put it here, how do cells know what to do? How do cells know what to do at the right time? One possibility is that the response of each individual cell is encoded genetically. We all like to talk about the genetic program, and this means that everything is in the genes, and the genes are the ones, there is a program there that is going to tell every single cell in that embryo what to do, depending on where it is at any given point, and what cells are surrounding it, and what chemical signals they are feeling at that point. But it is hard to believe that this can be encoded genetically. It is much simpler to believe that what is encoded genetically is the ability of the cells to build a representation of what's going on locally, which is changing in time, and then using this representation to behave in some sense. And this is our hypothesis right now. We have a complex environment. This complex environment acts upon cells, and cells should be able to create some internal representations that guide the behavior of this system. In this particular case that I was telling you about, for instance, these signals would come in the form of mechanical and biochemical signals, and then the behavior would be where the cells are going to move and how the cells are going to decide what to become. Okay, so for instance, motility and differentiation. And the question is, how does this happen? And of course, to understand this, there has been a lot of work being done for decades already at the level of brain. So I'm going to go back again to our higher order, to our neural systems, and there, these brains have this problem, right? And of course, maybe not so much at the level of neuroscience, but in psychology, for instance, in cognitive sciences, the idea that these brains are building internal representations of their environment, which are the ones that are guiding the behavior, is something that in psychology, this is one of the main concepts. So in some sense, we would like to use this analogy, the fact that you have high-level discussions of how the brain behaves at the level of psychology and lower level at the level of neuroscience, at the level of neural networks, to really link these two scales, the scale of behavior and the scale of structure, okay? Of course, we know, so how does this happen? What would be the analogy? We know that in the case of brains, this connection, this emergence of function from the structure should happen in terms due to communication between neurons at the level of electrical signaling. But of course, we don't know very well how this is happening because there is a huge gap between the micro and the macro. At the macro level, and then you have behavior. At the micro level, you have individual neurons in local circuits that you don't know exactly how to bridge. And this is a problem, as far as I understand, and you can correct me if I'm wrong, this is still a wide gap between the two fields. So the question is, how do you connect these two things? Well, we believe that maybe cells can serve as a useful model system because we also have networks of elements that are connected in complicated ways. No, for instance, at the level of gene regulation. So there is a clear analogy, and this is again what's driving the work in our lab. There is a clear analogy between neural networks in which you have neurons connected in complicated ways that are dynamical, and they are somehow processing information that comes from the outside and producing some kind of behavior. And the same thing would happen in cells, and of course at the level of the entire organisms, but what we are looking at is gene regulatory networks within cells. And there we do have also genes that can be activated or deactivated by other proteins coming from other genes, in the same way that neurons can be activated or deactivated by other neurons that are acting upon them. Okay, so this could be a basis for trying to make the mapping, and then maybe instead of using the brain to bridge the micro and the macro, using the cells to bring macro and macro, taking into account that gene regulatory networks are not as complicated in terms of the connectome, if you will, as the brain or as a neural network of a higher order organism. Okay. Now, a further analogy that I think is worth making is something that has happened recently. We know that these networks are very good at information processing because in the last 15 years, there has been this revolution, right, at the level of artificial neural networks. So, I mean, artificial neural networks were somehow inspired by real neural networks, and eventually people realized that just building multi-layer feed-forward neural networks of artificial elements are very good at classifying. And of course, lots of things have been done in the last 15 years that have eventually ended up in very specific architectures, such as the transformer, which is of course highly engineered. And this has shown that you can really do amazing stuff at the level of detecting structural regularities and behaving, if you will, by producing text like you do in large language models. I would like to point out, however, and I am happy to be corrected if you think otherwise. In my opinion, the way that we model artificial neural networks, in fact, is a better representation of what happens in gene regulatory networks than in neural networks, biological neural networks themselves. This is the typical model that you can see down here. And I don't know if there's This is the typical model of an artificial neural network. And there, what you have is you just have variables that determine whether the gene is on or off, depending on the inputs. This is ignoring completely the spiking behavior, for instance, of neurons, which maybe some computational neuroscientists and machine learning researchers are maybe considering, but this is a very fringe field, in my opinion. Most of the AI systems, most of the neural network systems right now are using this type of models in which they are ignoring spiking. They're just taking into account the fact that the units are on and off depending on the inputs. And I have to say, in my opinion, this is a more accurate representation of what's happening in genetic networks, because genes are going to either be active or inactive depending on their inputs. So in that regard, everything that we have learned in neural networks can be applied to gene regulatory networks. Now, the question is this something that makes any sense at all? What are the architecture of gene regulatory networks? And the advantage is that we have a lot of information about gene regulatory networks. We have databases such as, for instance, EcoSeq. This is the database of gene regulatory networks in E. coli, a very simple bacterium. And with this data, we know the genes, how they are connected with each other. So the connectome is pretty well mapped. There are around 4,000 nodes, which are connected by less than 10,000 edges. So this has been pretty well mapped. So now the question is, what is the architecture of this network? Is it following this kind of multi-layer feed-forward networks that work so well in artificial intelligence, because we know that these networks are really not such a good description of what happens in the brain to begin with, maybe in some sense, but this is a very abstract description. What happens here? Here we can do the analysis. Here we can really find out how everything is connected with everything. For instance, one thing that we can do is the following. Let me just give you a very brief example using just an abstract network. In here, we can consider that these are neurons or genes, whatever you want. So here you have a collection of nodes that are connected either in unidirectional or bidirectional way. And then what you want to know is what is the architecture. Is this feed forward? To what extent is this feed forward? Are there different layers? So what you can do very simply is try to establish how this is by looking at how the information propagates. For instance, one thing we can do is we can eliminate the nodes that have no inputs. No, we find the nodes that have no inputs, and this means that these are upstream nodes. So these are upstream from the network. So this would be the input nodes. We can imagine that the information enters through them. And we can do this, and then we can also do the same thing for nodes that have no outputs. And nodes that have no outputs, this means that this would be downstream. So we eliminate this, so this would be downstream nodes that are all of them connected without being connected with each other. They're receiving information from an upstream piece of the network. Now you can do this iteratively, because when you remove this node here, then now this one has no outputs. So now this one is also downstream. So then you do it like this, these two now are downstream. So they become part of a second layer. And you do this iteratively until you are not able to eliminate any node anymore, which means that here all the nodes have inputs and outputs, which means that all the nodes are connected with each other. So here there is nothing upstream, there is nothing downstream. Whatever is left are the different degrees of freedom of a dynamical system, of a nonlinear dynamical system. And the question is how large is the dimension of this dynamical system? Every node is connected with every other node. Okay, so now we did this in the network of E. coli, and we found this architecture. This is the architecture that we find if you apply this very simple algorithm to the network of E. coli that I was showing you before. What this is showing is at the very end, you have these nodes, these genes that have really no output. So these are the final layer, but then you have another layer of genes that are connected to the last layer, and so far, and so on, and so on, and so forth. So you can really see that most of the genes are connected in this feed-forward manner, which is great. But interestingly, there is a bunch of genes that are upstream from all of these that are connected with each other. Okay, and then of course you have the input nodes. Now we might ask, is this by chance? Is this what you would expect by chance? So now let me go back to this whole concept of network motifs that Uri alone defined 20 or 25 years ago, and ask you, is this a global network motif or not? If I just have a randomized version of a network, if I have my 4,000 nodes and I connect it randomly, what's the architecture that is going to come? Is this just by chance? And you can already tell that it's hard to think that this becomes by chance, because this core here is upstream of a huge number of nodes. Here you have around 3,000, as you will see in a minute, and here you have less than 100. And if this was by chance, at the very least, there should be a symmetry between nodes upstream and nodes downstream. Okay, and at most, the question is why there are so few of those. So if you drop these edges by chance in a network with the same number of nodes and the same number of edges, what you get is something like this. This is what you see here is the number of nodes in this, we call this a recurrent core, because this is a recurrent network. Okay, so the question is how many nodes we have in this core. In the case of E. coli, we have around 70. And how do we compare with the numbers that we would have in a recurrent core in a random network? And this is the comparison. This is the real network, and this is what happens in a family of random networks if we draw them by chance. Networks that correspond to structurally, they have the same number of nodes and edges than the original network. Okay, this is a zoom-in version of this. The important thing is that what we observe is very far away from both the mean of the distribution that we see in random surrogates and also the standard deviation. So the z-score is huge, which according to the definition of Uri alone, this would mean that this is some kind of global network motif. And the question is, why is this useful? Okay, so now why is this useful? One possibility is, let me compare how does information propagate in feed-forward and these recurrent networks. In feed-forward networks, information enters to the left, and then it proceeds towards the right, and eventually it's gone. So it's unidirectional propagation of information. You can imagine here that if the input is not stationary, and this works well when the input is stationary, when you have pictures of cats and dogs that don't move. But if the input is dynamic and these cats and dogs are moving, the information of what they are doing is escaping quickly. This network has no memory. The memory basically is given by the length that the information takes from, to go from the input layer to the reader layer, which is finite. Let me contrast with the situation downstream. If the network is recurrent, information can enter through the input nodes, and it starts reverberating, and this provides some kind of a memory, what we could call a fading memory to the system. Eventually, the information is going to disappear, but for a while, it stays here. And while it stays here, it can mix, it can combine the information that is remembering from the early past, well, from the recent past, let's say, okay, with the information that the network is receiving from the present. So this is a way for the system to process time-dependent information in a way that traditional feed-forward networks cannot do. Okay, I don't know if this is it okay? Anybody wants to say something that destroys my talk, and then I'll be happy to take it.
I think this is very clear. Jonas had a question, but it looks like it was resolved when you were talking about the recurrent code.
Okay, I'm sorry, Jonas. I didn't see your chat. Okay. Yeah. GRNs heavily rely on feedback. Yes. Well, in fact, then, as you can see, when we do the pruning, when we do the architecture, I mean, it's not so heavily in the. It's a bit surprising, because a lot of the nodes are in the downstream feed-forward network, which is also very interesting. Why is this the case? Okay. I'll come back to this in a minute. But anyway, thank you. Okay, good. So the other thing I want, Jonas, you go ahead. Yes.
Yeah.
Hi. Nice to see you.
Yeah. No. Like you were obviously getting there, so that's all good. Yeah. And just like maybe one thing that, since we're just on it, I'll mention, right? So those nodes that don't, like, it's not quite true, right, that there is no feedback from the output nodes from your output nodes to your input nodes, because that there is a layer through the environment which you're not capturing, right?
I love it. Yes, definitely. Yeah, I was talking about the structure of the network. The connections were exclusively gene regulation, but I agree with you. And not only this, of course, I'm ignoring other things. There could be, I mean, this is a network at the level of gene regulation, which operates on time scales of the order of tens of minutes or hours, depending on the cell type. But of course, there are other networks that we are also looking at right now. I mean, inside cells, which are signaling networks, metabolic networks. So the question would apply also for the same networks at different time scales. And this will for sure also provide feedback, but maybe the feedback is much faster. I agree with you, Jonas, that I think the feedback through the environment is more important. But I was just looking at the cell, and you will see why I'm focusing only on this, and I'm not thinking for the moment about the feedback from the environment. But anyway, Thank you. Okay, great, good. Okay, so yes, the other thing I wanted to say that it's somewhat important, and I think it's obvious, but I don't know. I just wanted to point it out. But I'm sure that many people are thinking in the same way. As the difference between the top and the bottom is that here. Basically, what you have is a dynamical system. This system can exhibit self-sustained dynamics, whereas the system on top cannot. The system on top is just going to react to whatever happens in the input. It's going to be driven by the input. If the input signal changes with time, the whole thing is going to change in time, reflecting what happens in the input. I mean, I'm not saying it's not powerful, but it doesn't have self-sustained dynamics. Whereas here, the trick here is that the information is going to come, and the connections you have here usually non-linear dynamical system of in our case 70 dimensions that can exhibit self-sustained dynamics. Here I have a very simple example. For me, as a very simple feed-forward system would be a classical feed-forward loop, which can be coherent or incoherent, whatever. And a classical example of a network of a network with feedback with recurrence would be a repressivator. And of course, if you compare these two things, if the feed-forward loop, if it's incoherent, it's going to respond to a simple step by just doing a derivative, if you will, and then going back down. So this system, it doesn't have a self-sustained dynamics, whereas this system does have a self-sustained dynamics. Simply, it's a dynamical system. It can be driven but it has its own timescales. And this is an important issue because we know from previous work by others, for instance, we know from work by 15 years ago and earlier work, this is a paper by David Cecilio and Larry Abbott from 15 years ago, that if you have a neural network which is recurrent, it's very reasonable if you have the right balance between excitation and inhibition. I'm not going to get into this today, that the system is going to operate in a chaotic regime, in a self-sustained chaotic regime, not a oscillatory regime that is a repressilator, but it can fall in a self-sustained chaotic regime like this one here. And what they show is that this type of regime is very efficient at performing tasks that are time dependent. So in this particular case, this is a very simple time dependent task that they have in their paper, in which basically the system has to perform this kind of periodic thing, seesaw, if you will, and then very simply, in a relatively simple manner, the system can reproduce this dynamical behavior after training. And you just have to train, for instance, in this particular case, the weights that go from the recurrent core to the output, and this will be an instance, if you will, of reservoir computing, so-called reservoir computing. And here you have some feedback that maybe could be what Jonas was mentioning, feedback from the environment that is basically being mediated by this output node. In any case, what these people and others have shown is that if the system has self-sustained dynamics, it has substantial information processing capabilities. So what we believe is that maybe what's happening in this network is that the system can process information in space and time, complex information in time, by taking up time-dependent inputs, encoding them here, and then using this huge feed-forward network, okay, to just interpret what's going on here. And you might say, why do I have so few nodes? Well, I mean, this is a 70-dimensional dynamical system. And my point is that in 70 dimensions, a dynamical system can do a lot. And maybe that's enough for the cell, for a simple cell such as bacterium, to really be able to process complicated information in a variety of ways. So basically, this is encoding these complex signals, and all these nodes here in the decoding layers are basically interpreting whatever this so-called reservoir is working on. So we have been working on testing this type of architecture using classical tasks. I'm not going to go too much into detail, but for instance, one classical task is just what we call memory task. So we ask the system to remember the past, and then we measure how far in the past it can remember for a given architecture. And then what we see in a standard measure is that it can get to FAT14, whatever that means. I'm not going to enter into this, but if the network was feed-forward, that means you would have a memory capacity, whatever, in a discrete time task on the order of 2 or 3 or whatever, depending on the number of layers. But here, very quickly, you can go up. And interestingly, what happens is that this critical memory capacity is maximum when this so-called maximal Lyapunov exponent of the system crosses 0. And as some of you may know, this Lyapunov exponent is a way of measuring when the system is becoming chaotic in a self-sustained manner. So when the Lyapunov exponents are negative, the system is not chaotic, it's regular, it's basically any information is going to decay down to 0. In this case here, as soon as the Lyapunov exponent becomes 0 and above, then here the system is going to be chaotic. And what we can see, and this is a classical result in so-called reservoir computing paradigm, near the situation where the Lyapunov exponent is 0, at the edge of chaos, if you will, this is when this system at least performs the best for a memory task. Okay, this will be the claim here. Okay, what happens is that on this area here, any perturbation that comes from the environment is going to decay very quickly. On this area here, so here basically in both cases, because here the same thing is going to happen. Here maybe the perturbations don't decay but stay forever. So here the system remembers a lot, if you will, and here the system remembers not too much and not enough. And then here you have this sweet spot in which the system remembers just enough so that it can process a signal. Okay, we tested other tasks. We can use, this kind of paradigm has been studied quite a bit in the machine learning field, even though I have to tell you that as soon as long short-term memory networks, LSTMs, and later transformers came into the picture, people forgot about this kind of computing paradigm simply because it's not as efficient as an LSTM or definitely not as a transformer. But in my opinion, transformers and LSTMs are over-engineered for nature. I think it's very hard to expect that evolution would have found a transformer inside a cell or even an LSTM. But what we are talking about here is basically a network that could as well be random. You just need to have a recurrent network that, in fact, it performs in principle better the more random it is. So that's the idea. So we have other tasks. We have tried other tasks, such as, for instance, asking the network to remember an input in the past and compare it with another input in the present, and then having this kind of delayed AND task, asking the network to really see, oh, if I have an input in this particular case, for instance, 6 time steps before, and I have another input that is high in the present, then you should respond with a 1, and if not, you respond with a 0. And you can see that very easily you can train this network to really follow the target very nicely. This is a just normalized root mean square error, and you can see that the system has a relatively low root mean square error. And even if you remove nodes, what we have done is also removing nodes out of this reservoir and making it smaller and see how the system performs. And there are situations such as somewhere in here in which even you can remove up to 30 nodes, and you can be left with 40 nodes in this network, and still you have a pretty good behavior, simply because as long as you have enough recurrence, you're going to be able to perform these tasks, okay? Now, we have done this analysis not only for E. coli, we have done it for other bacteria. This was not so good because the database was not so complete. We are repeating these studies right now. We have done it with yeast, we have done it with the fruit fly, we have done it with humans. These are coming from the genome, from the genome of these different cell types. And in all the cases, we found a reservoir which is on the order of a few tens or hundreds of genes which are connected in a recurrent manner, and that they are followed by a huge readout that in principle should interpret what's going later. For instance, here you can see in the case of human data, in the case of the human data set, which probably was not very complete at the time that we made the analysis. This is different from E. coli, that is quite well known. But in this case, we can see that the reservoir has 200 genes, followed by 13,000 genes downstream in the form of a feed-forward network. So we believe that this kind of architecture, I'm not going to go too much into this because I want to move on to some other example. This kind of architecture is just a way that the cells would have to really take a complex environment, encode it in an internal representation that is dynamic. So this internal representation would be a complex attractor, if you will, in the phase space of this 70-dimensional dynamical system. So this would be an instantiation of one of these internal representations that we still want to parse. We are working on this. And the idea next is to see how this thing is guiding behavior, okay? Anyway, this is one part of the talk. I would be ready to move on, but I forgot if these talks are supposed to be finishing. I mean, I could continue for 10 more minutes, but I'm not sure if this is appropriate. So I am.
We've got loads of time. We've got,
okay.
Yeah, very good in time. We've got still 25 minutes. You can fill them completely if you want.
Okay. Okay. I will not do it. I don't want to take all the time by talking myself.
But this is excellent. So like, take your time. We've got the time. And
okay. Anyway, again, so thank you, Leo. If anybody has a question, I would be happy to answer, or some comment. I would be happy to try to. But if not, let me tell you the following. So now I have to be honest with you. This idea, in my opinion, I think it's nice, but it's very hard to validate. And I'm sure that by now you have how do you validate this thing? How do you really do experiments? And that's what we are trying to do. We want to really validate this thing. But I mean, if a dynamical representation here is a strange attractor or a dynamical attractor in a 70-dimensional phase space, and how do we really validate that this is what's driving behavior in this case? So it's very hard. So we are working on this, and I have nothing much to tell you except that this is just a way of showing that maybe this could happen in cells. Cells have the machinery, the toolbox to really make this happen. Now, the problem here that we have is that if we are looking for internal state, so basically what would happen here, I haven't mentioned this, but another way of understanding what's happening here is that I already told you in some sense in passing, that what the cell is doing is mixing an external input from the environment, which happens at the present, with an internal state that has encoded the past environment. Okay, so the past environment has entered the network, it circulates, so the cell has information about the past, and it combines it with information from the present, which is coming directly from the environment through the input nodes. So there is a combination between the external state and the external input and an internal state. And this internal state can be interpreted in the form of this internal representation. Now, the problem is that right now, as I was telling you, the internal representation is very complicated. So if I want to validate the concept, maybe what I would need is a much simpler internal representation or a much simpler internal state. Okay, also maybe a simpler complex, a simpler environment, because I would like to parse out to distinguish between the internal state and the external input. Okay, these two things. So the question is whether we can find a life form with a simple enough internal state. Okay, to really validate this, because the problem with cells, this is bacteria, but it happens everywhere. I mean, the internal state of a cell is going to be very complicated in any way you want to look at it. I mean, I guess we can all agree that the internal state of a cell is going to be the combination of all the macromolecules and even all the molecules in general that the cell contains, and which is going to vary dynamically. And this is very complicated. And in part, it's complicated because the cells are continuously perceiving the environment. So the past is always there. So how do you separate? How do you dissect the input and the output? So I would like to tell you in the next, let me say 15 minutes, if I may. Hopefully I will not take more, and then we can take time for questions. I would like to present to you a system that we have been working on in the past and we keep working on, of a living system that has an extremely simple internal state. And it is, I don't think it is so common because of the reason I was telling you. As a cell is living and is doing its own things, it has to encode all these environments. So can we have something that is almost not living? Okay, I would like to have a life form that is almost not living. And the life form is a bacterial spore. Okay, so what I would like to discuss with you now, I would like to use this system, a bacterial spore. Okay, a spore is as close to an unliving organism as possible, because a bacterial spore, these bacterial spores are dormant, are fully dormant metabolically. As you probably know, maybe you know better than I. Basically, these are just hard coats surrounding, look, hearts. Anyway, hard cores that are surrounding a chromosome. Okay, that's basically what they have. They almost have no water. They have very little energy. They have almost no ATP. They cannot spend ATP, so they are dormant. Okay, this is the price they have to pay. Of course, what they win is that they can survive for thousands of years without anything. They don't need nutrients. They don't need anything. That's like they are like rocks, but live rocks that are alive. Okay, so this thing has a very simple internal state. So maybe here we can separate internal and external, the internal state from the external input. Now, I have to tell you, this is completely different from what I was saying earlier, because these spores are definitely not expressing genes, and therefore, here the cell is not relying on gene regulatory mechanisms, on gene regulatory networks to encode. They cannot. Okay, these spores cannot rely on gene expression, on dynamical gene expression, on regulating gene expression in time. So it has to be something else, something in fact simpler. That's what I would like to convince you of. Okay, let me just for those of you who are not very familiar with the spores, let me just show you a video that depicts some sporulation. Forget the colors. This is just a video that we took quite a few years ago, and what you see here is spores being formed. Spores are these white objects that are appearing at different times, coming from mother cells. The mother cells eventually die, giving rise to these spores, and these spores are extremely resistant. This is a microcolony of bacillus satellite cells that is very stressed, and they're basically the final stress response they have is becoming spores or dying. Okay, so this is the way that these spores are formed. So what the mother cell is doing is enabling this little spore to get rid of most of its water, becoming dehydrated, and surround itself with a very hard coat. Okay, so that's so far so good. So the spores are formed, and then the question is, how do the spores survive, and how do they come to life again? So because one of the things that happen is that these spores can be in this dormant state for thousands of years. So people have really obtained spores from soil, from terrain that is thousands of years old, and revive this spore in a few minutes, if you give them the right conditions, okay, the right food, if you will, the right signals. So this is just an example of how does this happen. So for instance, if I take a spore which is basically dehydrated, white here in this phase contrast image, white means no water or very, very little water. Okay, so when you see this thing white, it's because there's little water, and when it becomes black, it's because it has more water inside. It's bringing up water. Okay, so what I'm going to show you is what happens with this spore when you add a particular germinant, as it's called. In this particular case, it's L-alanine, an amino acid. You only need L-alanine, and if you add L-alanine to the spores, what's going to happen here? These are minutes. In just 10 minutes, you can see that the spores are going to become hydrated again. It only takes 10 minutes. I have to tell you, these things can be stable for thousands of years, even in water. Well, I don't know for thousands of years in water, but if they are in soil, and the soil is not very, I mean, in water, you can have them for months for sure. I mean, anybody who has spores in their labs, months or even years, you can have spores stored in water. Maybe, of course, you can freeze them and whatever. But in 10 minutes, they come back to life. They basically hydrate themselves in 10 minutes. Okay, so far so good. Now, if this thing has a simple internal state, why don't we apply a complex environment, but not too complex? Okay, because here the environment is very simple. There was nothing, and now I add some germinant, and I keep the germinant there, and I see what happens. What happens if I change the external signal and I make it dynamic? And one thing that we can do is we can take pulses of germinant instead of continuous germinant. So I just, for instance, in this particular case, you'll see what happens when we apply a 3-minute pulse followed by 2 hours of wait, 3-minute pulse followed by 2 hours. So we apply multiple pulses. Okay, and what happens in this case is seemingly nothing. Okay, you apply the pulses when you see the orange, you apply the pulses, and the spores seem to not respond. So this is kind of boring. Okay, what can we do? But let me tell you what happens when we look at this at a more high scale level. What happens if we do this experiment by looking at hundreds of spores, if you will, for instance? So we can do that by using a microfluidic chip. We place our spores here. These are experiments done by my collaborator Gulosuel at UCSD. I will mention him later because we have been working on this for many years, and I have a lot of fun working with him. So basically, you can place the spores in this microfluidic chip. You can flow in the nutrients or whatever. In this particular case, just water, and then every 2 hours, you just flow in 3 minutes of germinant, and then you flow it out. Okay, so you have a dynamical environment. And let me show you now how do these spores respond to this dynamical environment. So here, this is the picture, the movie, and here I'm just going to plot as time evolves, germination events. So I'm going to show, let's see if I can show it here. Oops, sorry, let me see. Okay, what I'm going to show here, every one of these lines is one spore. When you see it gray, this means that the spore is still a spore. And when it becomes black, it has germinated. Okay, so now at hour 1, we apply the first pulse. And you can see, pip, you see 3 that have already germinated. Now we apply again 1 at hour 3. Oops, oh my God, sorry. Okay, 1, 3, okay? So now many more. And you can already start thinking there is something weird going on. And I hope you agree. I mean, first of all, there was something weird here, which is, I think, nice, which is that just a few, probabilistically, some of the spores are going to respond. But then the interesting thing is that after 3 hours, the probability seems to grow. And we can continue doing this, and we can, and then we can see that eventually almost every spore in the dish or in the chip is going to germinate. And we can quantify this because we can do this for many, many spores. And what you get is the following. This is basically what I'm plotting here versus time is the fraction of spores that remain dormant. And after the first pulse, most of the spores remain dormant. In other words, and this is a different way of viewing it, the probability of germination is very small after the first pulse. But as you have seen, we didn't just quantify what you have seen. If we apply it on a second pulse, the dormant fraction really decays a lot. In other words, the probability of germination has gone up. So now, if the system, if these spores really were not doing anything, the probability of germination would be constant. I hope you agree with me. They would have a constant probability of germination. If the probability of germination increases a lot, from less than 0.1 to more than 0.5, from one pulse to the other, this means that something must have changed in the spore. Something must have changed with the spore between the first and the second pulse. Okay, so the internal state of the spore must have changed because of the external input. So here we have the two things and we can pinpoint them. Okay, so then the question is, what's changed? What has changed in the internal state of the spore between before the first pulse and before the second pulse in the two intervals? What has changed? Now, there are not too many things that can change. Gene regulation cannot happen. These spores don't have energy to express genes to do many things, so they cannot use processes that require ATP. So what we eventually stumbled upon is that maybe the most likely candidate is basically using ions, okay? Because ion flow through membrane can be a passive mechanism. Because since any cell, basically across the membrane, you are maintaining a gradient of concentration between these ions. The only thing that you need for the cell to change is opening a channel. And opening a channel can be a conformational change that requires, in principle, no energy, no? So in principle, if the spore is able to open a channel here, and then this means that the ion concentration inside and outside is going to tend to equilibrate, also taking into account the Nernst potential and whatever. I will come back to this in a minute. But then if you open the channel, ions are going to flow through the membrane. And this is going to be for free, if you will. Okay. And in fact of the matter, we know that most of the cellular content of a bacterium, for instance, in this case, in terms of number of molecules is ions. And most of the ions, the majority is potassium, for instance. And let's keep that in mind for later. Okay. Okay. Now we got to this hypothesis because together with Gurol, in the last few years, we have become convinced that electrochemistry is very important in bacterial cells. For instance, we know that potassium waves are able to coordinate stress responses in biofilms. Here you can see a wave of potassium propagating from the interior of a biofilm to the exterior, just to convey information of stress. Okay, so bacteria use these ions as a coin, as currency, or as a communication channel. Okay, this communication channel also can extend across colonies. So we have another paper in which we saw these bacterial biofilms oscillating in synchrony because of the coupling through ions. And we know it's through ions because if we knock out channels, this synchronization disappears. Okay, so this we know. I'm not going to go into details, but also bacteria use ions to stabilize ribosomes and therefore protect themselves against antibiotics. Okay, so we know that bacteria use ions a lot. And when we came to this situation, immediately, you know that when you have a hammer, everything looks like a nail, but I hope that this is more like that. It's more than that. Basically, we thought ions are playing a role here. Okay. In particular, one thing that we saw is that we know this is something that we know. It's not that we saw only; it's we know that potassium is higher in spores than in vegetative bacteria. So the intracellular concentration of potassium is higher in spores. That's a fact. Okay, so which is also interesting because it seems as if spores are somehow storing potassium more than they do when the bacteria are growing normally. Why is that? Well, the idea is the following. First of all, let me show you that these experiments that were done by Gurol and Kaito Kikuchi, I will mention them later. Basically, what we see is that if you track the extracellular potassium that these cells have, so right at the outside of the membrane of these spores, when you apply the germinant pulses, what you can see is that the potassium goes up slightly, but noticeably. Okay, so the cells are here in terms of how much potassium you have in the outside. You apply a pulse, potassium goes up for a while and it relaxes. Potassium goes up after another pulse, potassium goes up for a while and it relaxes. If you use a channel blocker for the potassium channel that we believe is playing a big role here, basically this thing goes away. Okay, so indeed, cells are releasing potassium in response to these germinants. So what we can say is that maybe the internal state of these spores is basically stored in potassium, in the intracellular potassium concentrations. And then what you have is changes in the ion concentrations inside and outside due to this opening and closing of the channels, depending on whether the germinants are affecting or not affecting the cells. So if that is the case, then we can build a model. No? Well, I mean, of course, and now we go to this is a non-neural similar series, but now I go to my neural counterpoint. I go back to the analogy between cells and neurons. and brains, if you will. But of course, neurons operate because of the flow of ions across the membranes, potassium, sodium, calcium, etc. No? For instance, the classical model of neuronal electrophysiology, which is a Hodgkin-Huxley model, basically contains models a channel of a current of sodium, a current of potassium, and the rest of the ions moving through, which is a leak current. Okay, so what we can do is we can do a simplified version of this model in which we have only the potassium current, because we see that sodium doesn't play any role. This is data that I'm not showing you today, but sodium doesn't play any role here. So we have the potassium, and then we have a leak current, and we can build a model that basically reproduces. The idea would be whether it reproduces the behavior. Now, since I'm already quite advanced in my time, I'm not going to show you. The model is basically you just have intercellular potassium and extracellular potassium, and then you just have a channel that can open, and we can model the membrane potential dynamics. And this is very much inspired by Hodgkin and Huxley, the Hodgkin and Huxley model. So the whole thing is driven by the Nernst potential of potassium. And then what we have on top of that is this is going to also change the concentrations of potassium inside and outside. Okay. So basically, and then with this model, we test the following assumption. The idea is that the germination propensity is inversely related to how much potassium the cell has, the spore has. At the beginning, the spore has a lot of potassium. When it receives germinant, if it opens up, it starts releasing potassium, and eventually it reaches a threshold. And when the potassium levels are low enough, then the spore is going to germinate. Water is going to come in, and then the cells are going to be revived. Okay, this is the model. Okay, so sorry, I didn't want to show you this, but I want to show you this. This is the version of the model. So it's all determined by how much potassium the spore has at the beginning, the efflux of potassium. This determines the dynamics. So this would be similar in a much lower scale to the dynamics of our huge gene regulatory network. So in this case, we just have a single degree of freedom, which is how much potassium the cell has, which is dynamic. And this thing is going to determine this is going to determine behavior, which is propensity of germination. Now, if we apply this model, we can obtain very easily, without a lot of fine tuning of the parameters or anything, very reasonable parameters, we can obtain results from a simulation of the model that very closely mimic the experiment. Of course, we are still at tuning parameters, so you could say, what's up with that? So what we can do is we can validate the model by playing with mutants. So what Gurol had in his lab is an amazing set of mutants, an amazing set of people that allowed him to do mutants in which we basically eliminate a pump that changes the initial concentration of potassium, that eliminates the channel, that changes the ability of the ions to get out, or block the channel. And this is a chemical versus these genetic perturbations. And we can make predictions that validate this model. I'm not going to go into this because we are almost out of time. What I was just to tell you is that this is the model of what's going on. It's very different from the first part of the talk. It's much simpler. And what I'm telling you is that we believe that what these spores are doing they are following what we could call an integrator fire model, which is a typical model for neuronal dynamics, in the sense that the cell is integrating pulses by releasing potassium, and at some point they reach a threshold and then they fire. It's the same way that neurons do. They integrate action potentials from presynaptic neurons, and at some point they're going to surpass a threshold and then they fire. In the case of neurons, firing means spiking. In the case of spores, firing means germinating, and you can only do it once. But still, the idea is the same. So this is what we wanted to propose. This is basically the idea. I'm almost done. I just wanted to tell you that the final thing that we can do is that if we really want to mimic let me tell you here, well, I have it here. If you go to the model, you can see that we have a membrane potential. That's what we have in the Hodgkin-Huxley model. What about the membrane potential of the cell? A prediction of the model is that the cell is going to change its membrane potential when the germinant comes. So you remember the video I showed you at the beginning? Germinant pulses come and nothing seems to change in the spore. Nothing seems to change. More hearts to all of you. Anyway, nothing seems to change. So let me show you now what happens, because what we are assuming is that as the cell loses potassium, its membrane potential it's going to grow, in the sense that the cells are going to become less and less negative. No, you lose it, so you know that cells are negatively charged. If they keep losing potassium, positive charges, they're going to become hyperpolarized stepwise. This is the prediction of the model. And we have a dye that we can use to really measure the membrane potential, which is theoflavin T, which is a dye that basically is going to stain the spores when they are hyperpolarizing. The more they hyperpolarize, the more THT they are going to take up. And what the model says is this. We can model it very easily. And here you can see the experimental results in which the different spores, whenever they see when they receive a germinal pulse of 3 minutes, they're going to jump in their electrochemical potential. They hyperpolarize. They receive another pulse. They keep hyperpolarizing. Of course, this one no, because this one germinated already, but this one continues. And then this one now germinates, the red one, and the yellow one keeps growing. Okay, and this is a video in which you can see this thing happening in vivo. Okay, so here you see the different cells that they keep growing in membrane potential as they receive the membrane. So this is an indication that the internal state of these cells is really responding to this relatively complex, not so complex, but still dynamical environment. So it's changing its state, and it's incorporating the current state with the previous state in a very simplistic manner, at least from our understanding. But this leads to the response of the system. So anyway, this is my take-home. These are my take-home messages, and these are different. I mean, this is maybe mostly for the first part. The second part that I showed you is basically an instantiation of the fact that you can have a very simple internal representation in the form of just potassium concentration. But the main message I wanted to transmit to you is that biological networks are commonly recurrent. The recurrence is not widespread. Is quite structured, and this network architecture allows for information processing of time-dependent signals, and this dynamical state provides an internal representation of the complex environment of the cell. Now, let me finish with thanking people. This is the people of my lab that have been working over the years on this topic. The latest one is Maria, who is here. Many of them are not in this picture because they already left the lab quite a long time ago. But I would especially like to thank my colleagues at UCSD, Gurol Suel, a colleague and a friend. I've been working with him for 20 years already and having a lot of fun. And he has an amazing group of people that have been involved in the things that I have been explaining to you in this in the second part of the talk. So thank you so much.
Wow, thank you very much for this very inspiring talk. That was brilliant. I think
thank you.
Maybe it makes sense first to give the audience the possibility to ask questions. Absolutely. So please, if you have any questions, please unmute yourself, or if you're feeling shy, then there's no problem. You can text in the chat and I will read it out loud. But maybe to get this going, I can start with an initial question. Oh no, Oliver, here we go. I see you. You unmuted yourself. Please go ahead.
Yes, I had one question, Jordi. Excellent talk, as usual.
Olivier, hello.
That's me.
So long. Nice to see you.
Sure. Regarding the first part, the GRN part, to which extent your recurrent core is what we would call the low-dimensional manifold on which the cells should be living? I mean, is there any kind of a correspondence there?
That's a wonderful question. I never thought of it in this way, in the sense that for me, I like to think that it's relatively high dimensional so the dimension is high enough for what the cell might need. Yeah. But still, I agree with you. So in some sense,
much lower. I mean, 200 for humans is much less than 20,000, right?
That is true. That is true. No, that's a wonderful question. I agree. I agree in some sense that it's some kind of a dimensionality reduction of the entire genome for a certain type of concept. I mean, this doesn't mean that this is the response to the explanation for everything. But at least for these particular tasks, I agree with you. Thank you.
And my other question would be, like each node is a gene. So it's a dynamical system per se, and it's a probabilistic dynamical system. Did you try to see if the nodes become probabilistic? Does that change anything in the way they process information? Would that help them remember or just remove some kind of a memory, or what impact might it have?
That's a wonderful question. To be honest with you, this is something that we wanted to test and we have been working on for a while, but very slowly. But definitely, this is something that we want to look at. For the moment, to be honest, the models that we are using are still, in some sense, not always. I mean, it depends on the project that we are working on. But the models that we are using are mostly typical machine learning models, which are discrete in time, with sigmoids and whatever. Yeah, but the question is super interesting. I agree with you. And yeah, you know that we love noise, right? So
yeah, I know. That's why I would ask the question. Noise forever. Okay, thanks a lot, Jordi.
I'm so happy to see you, Olivier.
Me too.
Okay.
Bye-bye.
Bye. See you in a minute.
Brilliant. I can see that Oliver and Sylvia both have another question. Like in my chat, Oliver was first. So Oliver, please go ahead.
Hi, thank you very much for the talk. So I'm a chemist, not a computer scientist, so you'll have to forgive me for slightly ignorant question. But my understanding of reservoir computers was that you sort of the training is done on the output in order to interpret what's going on or to pull out whatever kind of representation you're trying to do or learning. So what is the training mean in this here in the cell in the gene regulatory network? What's actually going on? Is it how are you kind of selecting different outputs? Is it certain expression of certain transcription factors or what can you shed perhaps a bit of insight there?
Yeah, that is a wonderful question, and yeah, I'm always very respectful of chemists, which I so
we have dangerous chemicals.
So no, apart from that, yeah. So no, it's a great question. So I haven't gone through this in the talk. There are different ways of looking at it. I mean, my first answer would be, look, we are looking at gene regulatory networks. I agree with you that in the simplest perspective, in the context of reservoir computing, a different thing is in a general recurrent neural network context in which Susilo and Abbott have been working on, and they are in some situations they train the entire network, including inside the reservoir, and we are also doing it in the lab as well. Okay, but the reservoir computing paradigm is really attractive because, as you say, you only need to train the weights, the connections that go from the reservoir to the output nodes. And definitely we have been working on this for a long time. And in this regime, this is where the fact that you have a Lyapunov exponent around 0, this corresponds to a maximum critical memory capacity, because we are in the conditions in which reservoir computing is operating. So that's true. Now, what about learning? So of course, machine learning researchers basically train these things in a very simple manner. So in fact, these networks are super attractive for machine learning researchers because you can train them with a simple algebraic operation since you only have to train the last weights. This is very simple, but of course, cells are not going to do that. So one of the things that we did in the very first paper that we published on this quite a few years ago is to forget about this way of training that is very artificial. And we were thinking, look, the simplest way in which these weights can be trained, which at the end of the day is promoter activities, right? So the effect that a transcription factor has on a promoter. So I mean, the simplest way would be maybe evolution is acting upon this by changing the sequences, and by changing the sequences of the promoters, for instance, you can tune the weights. Of course, this would be very slow. I mean, but this is the idea would be that cells evolution has really trained this type of networks and set up a set of weights that enable these cells to develop the internal representations. Cells do not need to learn these things from scratch every time. This is program, if you will. This is program in the genome. The ability, the reservoir computing ability is what is programmed in the genome, if you will. And of course, another option is other things, as you were saying, other things could happen. I mean, cells could regulate the abundance of their transcription factors that in a different the activity. Sorry, cells could regulate the activity of the transcription factor by using metabolites or in other ways, so that they can really learn online. But this is not. We are not considering this right now. We know, because for instance, if you apply genetic algorithms, this is something that we have done. You can train these things very easily, maybe a little bit more slowly, but still very easily. You can train them with genetic algorithms that mimic what evolution is doing, if you will, in some sense. This is not as efficient as using these algebraic methods that machine learning researchers use. They do it. You can do it instantly. Here, you need a few thousand generations of your evolutionary algorithm, but then you can. I don't have, or maybe I do have the data here. Let me see if I can. Here. You see here. So this is a comparison of the training between what the machine learning researchers use, which is called rich regression, which is an algebraic operation that gives you the weights of the readout nodes, versus doing an evolutionary algorithm in which we just mimic what evolution is doing. And after a few generations, we get to the same thing. So our interpretation is that these cells have learned over their evolution to really perform certain tasks. I think the nice thing is that if they need, the nice thing about reservoir computing that I'm sure you know is that if they need to learn a new task, they don't need to train everything again. You just add new readout nodes. That's the beauty. I mean, I'm really a bit obsessed, and maybe unnecessarily so, with this paradigm, because it's very beautiful. If the cells encounter a new task, they don't have to relearn everything. They just create a new readout node or a new set of nodes, and they train these nodes. And after a few generations, now the cells can perform this task. So my long answer to your question is that in this case, for the moment, it's evolution, but there could be other methods, of course. Yes, thank you.
Okay, thank you.
Excellent. Maybe a last very quick question from Sylvia. Please go ahead.
Thank you. Nice to see you, Sylvia.
Yes, nice to see you as well. So I had a question on the spores. So if I think about a reaction of a neuron to the polarization of the membrane, I would expect this to happen on a very fast time scale. And the data, actually, the next slide, they seem to show that the response in these populations takes some time. Why is that?
Okay, that is a wonderful question. Maybe I'm going to show you this other thing that we had, because for instance, this type of behavior. If you look at this, again, it's the same idea. I mean, it's the same ion, it's the same channel. Here it's acting on vegetative cells. In the other case, it's acting on spores. But you can see here that, again, the time scale is very slow, to be honest. I mean, and the same thing is, so we are talking about time scales of tens of minutes, hundreds of minutes, or something like this, which is very different time scale from neurons. Now, why is this happening? Well, we believe this is an ancestral, I don't know, what we would like to think is that this is an ancestral behavior that maybe neurons really adapted, because here what we only have is you only have one ion. You only have one ion flow. You don't have the counter ion, which would be sodium. So in the case, for instance, if I go to the simpler neuronal system, there you have potassium and sodium, and the interplay between them can generate things that happen in the millisecond. Here, you just have a channel that, in fact, in the case of what you're seeing here on the slide right now, this is triggered by stress, and this is very slow. But this is the time scale that these cells need for this particular thing, because it's a time scale of growth. So it's 100 minutes, and this is enough. But I agree with you, there is this difference. But we like to believe that maybe evolution really picked up on this to really make things more complicated and eventually get into the neurons and find, if I play with another channel that has maybe more complicated structure, maybe I can get something faster. Yeah. Thank you.
Okay, thanks a lot.
Thank you. So eventually, I'm sorry that I go back to neural systems. So maybe, hopefully I hope you don't kick me out of the...
Absolutely not. No, that was brilliant. And obviously, that link makes absolutely sense with the Hodgkin-Huxley model that you presented as well. I think there are no other questions in the audience, at least not that I can see.
I mean, I have about a dozen questions for you, Jordi, but I don't know if we are out of time, so...
Well, I mean, if we are or whatever, I mean, maybe we can talk offline or we can meet another. I don't know if you're going to visit here, because I know that you have a collaboration going on with James.
Yeah, I'm going to be there in November. Yeah.
Well, please write to me and let's meet.
Will do.
That.
Let's do that. Okay. Cool.
Wonderful. Okay. Wonderful. Thank you.
Jordi, that was wonderful. Thank you so much for being
with
us today, for presenting your brilliant work. That was absolutely fascinating. I can only agree with the others that, I've got millions of questions now coming up, so loads to think about and food for thought. With no further ado, because we are already 15 minutes late,
sorry.
Thank you so much for coming. To the others, please tune in also to our next lecture as well, which will be in a month. All the updates will be posted in the group, and was a super It was a really great presentation. Thank you so much, Jordi, again. And
Thank you. I'll try to join again as an audience. Thank you.
Hopefully see you soon. Bye-bye.
Bye-bye. Thank you so much. Bye, everyone. Thank you.